Okay, so let's unpack this. If you work in oncology or, even if you just obsessively follow biotech headlines like I do, you'd know the dogma. For the last decade, the story of cancer immunotherapy has been a single character drama.
Yeah, the T cell.
Right, the hero is the T cell, specifically the CD8 positive cytotoxic T cell. We have built, entire pharmaceutical empires on this one specific cell.
It is absolutely the prevailing narrative, the assassin, right?
Why?
The idea is really simple. The tumor puts the brakes on the T cell, then drugs like Keytruda or Opdivo come in and cut those brakes, and the assassin wakes up and engages in hand-to-hand combat with cancer.
It's simple, it's elegant,
And it won a Nobel Prize.
Exactly, it won a Nobel Prize. But here is where it gets really interesting for our deep dive today. We have a new paper in front of us, published in Nature Medicine in early 2026, that completely flips that script. It suggests that the soldier might actually be totally useless without the artillery.
Yeah, this is a mind-blowing study. It's titled Humoral IgG1, Responses to Tumor Antigens Underpin Clinical Outcomes in Immune Checkpoint Blockade. It is a bit of a mouthful, I know, but the implications here are just massive.
Huge.
It suggests that while T cells might do the actual killing, the outcome, whether a patient actually lives or dies, might depend entirely on a completely different cell, the B cell, and specifically the plasma cell.
So if the T cell is the infantry charging in with bayonets,
Then the B cell is the heavy artillery shelling the enemy lines from a distance. That is the perfect analogy. And as any military historian will tell you, if you send the infantry in without artillery support, they just get slaughtered.
right.
This paper represents a fundamental shift in our understanding. It's a deep dive into hepatocellular carcinoma, which is liver cancer. But they didn't stop there. They validated this across melanoma, lung cancer, and over 1,500 patients.
Wow. So our mission today, for you listening, is to go basically sentence by sentence through this discovery. We are going to figure out how these antibody factories work, why they're the literal difference between life and death, and how they might change how we treat cancer forever.
Absolutely. And to set the stage, we really need to define the stakes here. The researchers looked at a cohort of 38 patients receiving neoadjuvant anti-PD-1 therapy.
And neoadjuvant means they got the immunotherapy before the surgery to actually remove the tumor, right?
Correct. This is the perfect time to look at what's happening because you can see the immune system's initial unadulterated reaction to the drug. And they divided these patients into two groups: responders and non-responders.
Let's define those for the listeners. A responder is someone whose tumor was, more than 50% dead when the surgeons took it out?
Yes, greater than 50% tumor necrosis. The drug worked. A non-responder is someone whose tumor basically just ignored the drug and kept right on growing. And the mystery has always been, why? Why does the exact same drug work miraculously for one person and do absolutely nothing for the next?
And usually researchers just look at the T cells and say, oh, they must be exhausted. But this team, they took a totally different approach. They didn't just look at a slice of tissue under a microscope. They performed single-cell RNA sequencing on a massive scale.
They did. They analyzed 1.2 million transcriptomes.
Okay, let's pause on that word, transcriptome. Because that's not just, counting cells.
No, not at all. If the genome is the instruction manual for the cell, the transcriptome is the diary of what the cell is actually doing right now. It tells you exactly which genes are turned on and which are turned off at that exact moment.
That's a great way to put it.
Yeah. So by looking at 1.2 million transcriptomes, they were essentially reading the diaries of every single immune cell in these tumors.
And from that mountain of data, they isolated about 30,000 specific B cells and plasma cells. And this is where the plot twists. What did they find when they actually compared the winners and the losers?
The difference was just stark. In the non-responders, the patients who didn't get better, the B cells were really underwhelming. They were mostly what we call dysfunctional memory B cells, or cells expressing IgM and IgD isotypes.
Okay, we got to get into the weeds here. IgM and IgB. For the listener who hasn't opened an immunology textbook since college, what does that actually tell us about the cell?
Think of IgM as the default setting. It's the very first antibody a B cell makes when it's immature or just starting out.
Yeah.
It's low affinity. It is not a precision weapon. It's kind of like a rookie scout wandering around the battlefield with a blurry map. It hasn't really committed to the fight yet.
So the non-responders have these confused rookie scouts wandering around. What about the responders?
The responders were packed with IgG1 positive plasma cells. Now, a plasma cell is the terminal stage of a B cell's life. It has stopped exploring, it has settled down, rooted itself in the tissue, and turned into a dedicated factory.
And specifically IgG1 antibodies, why does the one matter there?
So the IgG class has different subclasses. IgG1 is the heavy hitter. It has a specific tail region, the Fc region, that is incredibly good at signaling other immune cells to attack. It's the loudest alarm bell the immune system has. And genetically, these cells were just lighting up. They were expressing genes like MZB1, J-chain, and IgHG1.
Okay, let's break those down. I see J-chain and I think of like a chain link fence, but that's not it, is it?
Not quite, but you're somewhat on the right track with the linking idea. J-chain stands for joining chain. It's a protein that is critical for assembling and stabilizing these massive antibody structures before they are secreted out of the cell. If a cell is expressing really high levels of J-chain and MZB1, it tells us that cell is in overdrive. It is manufacturing proteins at an industrial scale.
So the big aha moment here is that we used to solely look for cellular immunity, the T cells. But this paper says humoral immunity, the fluids, the antibodies, is statistically much more predictive of who actually survived.
Yes, the T cells might pull the trigger, but the plasma cells are identifying the target and supplying the ammo. If you go back to the battlefield analogy, the non-responders have confused scouts. The responders have set up these heavy artillery stations right inside enemy territory.
That is such a powerful image, but here's the part that really twisted my brain when I was reading the methodology. Did these factories just appear out of nowhere? Like, did the drug create them from scratch?
That is the million-dollar question. Is it nature or nurture? To answer it, the researchers use something called clonal tracking.
Ah, this is the spy novel part of the study.
It really is.
Yeah.
Every single B cell has a unique barcode. It's CDR3 sequence. It's a random reshuffling of DNA that happens when the B cell is first born. If you find two cells with the exact same barcode later on, you know they're clones. They came from the exact same parent cell.
So they tracked these barcodes, and what did they find?
This is crucial. They found that the specific B-cell clones that helped cure the patient, the ones making that powerful IgG1, were already there before the treatment even started.
Wait, so the cure was already inside the patient?
Yes, they were pre-existing.
Yeah.
But they were dormant, or just incredibly low in numbers. They were sleeper agents. But the moment the patient received the PD-1 blockade, the drug, these specific clones woke up. They underwent massive clonal expansion. They just exploded in numbers.
So the drug is literally the trigger code that wakes up the sleeper agent.
Exactly. But there's a whole geography to this, too, which is fascinating. These clones weren't just sitting in the tumor. The researchers found the exact same barcodes, the same cells, in the draining lymph nodes.
The lymph nodes are like the training camps, right?
Correct. The data suggests a trafficking loop. The B cells are likely encountering the antigen in the tumor, realizing, hey, this is bad, and then traveling out to the lymph node. In the lymph node, they undergo training. Specifically, they switch from that weak IgM to the powerful IgG1. Then they commute back to the tumor to set up shop as a plasma cell.
Wow. And the non-responders, what happens to their cells?
Their B cells never make the switch. They stay as IgM. They likely never complete that training loop between the tumor and the lymph node.
Okay, let's do a quick recap here, like a 5-minute summary, because this mechanism is super complex and I want to make sure we're tracking.
Good idea.
Number 1, T cells aren't the only heroes. You absolutely need these IgG1 positive plasma cells. Number 2, responders have high IgG1. Non-responders have low-quality IgM or IgD. Number 3, these cells are pre-existing. They are sleeper agents waiting for the drug to activate them. And number 4, they commute between the tumor and the lymph nodes to get upgraded.
That is a perfect summary.
So here's the question for you listening right now. If these cells are factories pumping out millions of antibodies, what exactly are they manufacturing? Like, what are they shooting at?
That is exactly the mystery the researchers had to solve next. And to do it, they used seromics, which is analyzing the blood serum to find the target.
And the target wasn't just random cellular garbage. It was something very, very specific.
It was. The IgG1 antibodies in the responders were specifically targeting cancer-testis antigens, or CTAs.
Cancer-testis antigens. The name implies they belong in the testes.
They do. Specifically, we're talking about proteins with names like NY-ESO1, MAGE-A, GAGE7, and PRAME. Normally, in a healthy adult, these proteins are only found in the testis.
And the testes is an immune-privileged site, right? The immune system isn't supposed to be looking there at all.
Exactly. The immune system is strictly programmed to ignore the testes entirely. But tumors are chaotic. Their DNA is mutating, it's unspooling. Often, liver tumors accidentally turn these testis-only genes back on, so suddenly a liver cell is expressing a protein that belongs strictly in the testes.
It's a foreign flag. It's wearing an enemy uniform that shouldn't be anywhere near the liver.
It is the perfect target because it's not on normal liver cells. It's only on the cancer. And the data here is striking. They showed that 63% of the responders had specific IgG1 antibodies against these CTAs right in their blood. Only 17% of non-responders did.
That is a massive difference. So the B cells are seeing this NY-ESO1 flag and just launching missiles at it. But how does that actually help the T cell do its job?
This brings us to a really elegant mechanism known as opsonization.
Which sounds like a cooking term, but I know it's not.
Well, you're not entirely wrong. It actually comes from the Greek word for seasoning or preparing for eating. The antibodies physically coat the tumor cells. They cover it. This tagging makes the tumor cell highly visible to the innate immune system cells like dendritic cells and macrophages. These cells essentially eat the tagged bits and present them directly to the T cells.
Oh, I see. It's like laser designating a target for an airstrike. The T cell doesn't have to search blindly in the dark. The B cell has already painted the target bright red.
Precisely. And there was a specific clinical patient story in the paper that proves this exact sequence. They had a patient where they found the NY-ESO1 antibodies in the blood before the treatment started. But the T cell killing, which they measure by a chemical called interferon gamma, only started after the treatment began.
So the B cell primed the T cell, the artillery softened up the target, and then the infantry went in?
That absolutely appears to be the sequence. The B cell response precedes the T cell attack. If you don't have the B cell tagging the target first, the T cell often just walks right past the cancer.
This is just fascinating. But I want to shift gears and talk about where this is happening, because the paper introduces this concept called spatial transcriptomics.
Yeah, think of this as Google Earth for the tumor. Standard sequencing just gives you a list of inhabitants. It's a census. It tells you we have 50 T cells and 20 B cells. But spatial transcriptomics tells you exactly who is neighbors with whom.
Right, who is actually sitting next to whom at the dinner table.
Exactly.
Yeah.
And in the responders, they found these IgG1 positive plasma cells hanging out in what they called immune-rich hubs. They weren't just sitting alone. They were surrounded by T cells, fibroblasts, and vasculature.
A bustling neighborhood.
Very bustling. And they were actively talking. The data showed high levels of signaling via IL-4 and IL-13. These are cytokines that specifically support B cell growth and activity. It's a highly cooperative workshop. The fibroblasts build the physical structure, the T cells protect the perimeter, and the plasma cells sit in the middle and pump out the ammo.
And the non-responders, what does their neighborhood look like?
A completely different landscape. The authors called it a stromal memory-exhausted reservoir.
Or essentially a desert.
A desert is a great way to picture it. The dominant cells there were regulatory T cells, or Tregs and monocytes.
The fun police of the immune system?
Basically, yeah. Tregs damp down the immune response. And the monocytes there were actively sending suppressive signals. So the B cells trapped in these regions were atypical or exhausted. They weren't making antibodies. They were just stuck in this terrible bureaucratic office where nothing gets approved.
So location really matters. You need a hub. You can't be in a desert.
You need the architecture of cooperation. If the plasma cell is isolated in a sea of Tregs, it just cannot function.
Okay, let's do our second recap here, right around the 10-minute mark, because we are getting deep.
Let's do it.
Number 1, the antibodies are targeting forbidden antigens, things like NY-ESO1. Number 2, this tagging the opsonization helps T cells wake up and see the threat. Number 3, location is everything. Success requires hubs where plasma cells and T cells talk via IL-4 and IL-13. And number 4, failure looks like deserts filled with suppressive monocytes and Tregs. So here's the interactive question for you listening. We know this whole process works in liver cancer, but is the liver just special? Does this apply to other cancers? And more importantly, can we engineer this?
That leads us perfectly into the validation phase of the study, because the authors definitely didn't stop at liver cancer.
They went looking for the universal truth. What other cancers did they check?
They looked at melanoma cohorts that were treated with PD-1 and CTLA-4 blockade. They also looked at non-small cell lung cancer in the famous Poplar and OK trials.
In the theory holdup.
The pattern absolutely held. High expression of the gene IgHG1, which is the gene that makes the IgG1 antibody, was significantly associated with improved overall survival across the board.
Across over 1,500 patients. So this is a universal rule of immunotherapy.
It really seems to be. It worked for PD-1, PD-L1, CTLA-4, and even VEGF-A blockade. But, and this is a highly critical exception, it only applied to immunotherapy.
Oh, what happens with standard chemotherapy?
Nothing. In patients treated with chemotherapy alone, high IgG1 levels does not predict survival at all.
Well, I guess that makes sense, right? Chemo is basically poison. It just kills rapidly dividing cells. It doesn't care about antibodies or immune hubs.
Exactly. Immunotherapy relies on the body's own natural mechanisms. Chemo just nukes the site from orbit. So this mechanism we're discussing is specific to how the immune system organizes a biological defense, not how a toxin kills a cell.
Okay, so we have the mechanism. We have the validation across multiple cancers. Now comes the part I love. How do we hack it? Because if I'm a patient, I don't want to just cross my fingers and hope I have the right sleeper cells. I want to make sure I have them.
This is where the researchers dug into the why. Why do B cells become plasma cells in responders, but fail to do so in others? They looked at the gene expression pathways to find the master switches.
And they found two genes that sound like, I don't know, corporate stress management consultants. ORIN1 and XBP1.
That is surprisingly accurate.
Uh-huh.
ORIN1, which stands for endoplasmic reticulum to nucleus signaling 1, and XBP1 are the master regulators of something called the unfolded protein response, or UPR.
Okay, unpack that for us. The unfolded protein response.
All right, imagine a factory that suddenly gets an emergency order to produce 1 million units a day. The machinery heats up. Things start moving too fast. Proteins literally get tangled. Stress builds up in the endoplasmic reticulum, which is the specific part of the cell responsible for folding proteins into their proper 3D shapes.
So the assembly line is jamming up.
Exactly. And if the cell can't handle that stress, it dies. It self-destructs. ORIN1 and XBP1 are the crisis managers that rush in, expand the factory floor, add more cooling systems, and ensure the protein folding machinery can actually handle the massive load.
So if you don't have ORIN1 and XBP1 turned on, your factory basically burns down before it ships a single antibody.
Essentially, yes. The B cell physically cannot become a high-output plasma cell without these genes turning on. It just cannot handle the volume of antibody production required to fight the tumor.
And what turns them on? What are the signals that tell those managers to get to work?
The study identified IL-6 and TNF tumor necrosis factor as the key signals that tell the B cells to differentiate and activate that stress response. And CXCL13 is the magnet, the chemokine, that pulls them together into those hubs we talked about earlier.
So here is the billion-dollar idea, the grand proposal for the future. We don't just give PD-1 inhibitors blindly anymore. What do we do instead?
We move to rational combination therapy. The logic chain the authors propose is brilliant, and it's highly personalized. Step 1. Screen the patient. Do they have pre-existing IgG1 clones against CTAs, like NY-ESO-1?
And if they do?
Great. Give them standard PD-1 blockade. They already have the sleeper agents. The drug will wake them up. You are good to go.
But what if they don't? What if they are a non-responder profile with 0 sleeper agents?
Then giving them PD-1 alone is likely a complete waste of time. You need to create the sleeper agents first.
How do you do that?
Vaccines. You could vaccinate the patient against these CTAs, like literally inject them with an NY-ESO-1 vaccine to force their body to create those specific B-cell clones. Then, once the clones exist and are waiting in the lymph nodes, you give the PD-1 drug to expand them.
That's incredible. Are there other options?
Or you use IL-6 agonists or TNF pathway activators. You artificially provide the factory's startup signals to help their stubborn B cells differentiate into plasma cells.
That is a complete rethink of how we treat these patients. Instead of just throwing Keytruda at everyone and hoping for the best, we check their artillery status first.
Exactly. It moves us from a one-size-fits-all approach to true mechanically driven, personalized medicine. It actually explains why so many clinical trials have failed in the past. We were trying to wake up an army that simply wasn't there.
This has been an incredible deep dive. We started with the T-cell soldier, and we found out the soldier is basically helpless without the B-cell artillery factory.
It really changes the entire paradigm. We have moved from a T-cell-centric view to a humoral cellular axis. You need both. You need the antibody factory to guide the assassins.
So as we wrap up today, I want to leave you with a provocative thought. We currently spend millions of dollars and put patients through painful, invasive procedures to get tumor biopsies. We slice up the tissue just to see what's happening inside.
This study shows that the key to survival, those anti-NY-ESO-1 antibodies, is floating right there in the blood.
Exactly. If B cells are this important, are we currently ignoring the most powerful biomarker we have? Could a simple, cheap blood test for NY-ESO-1 antibodies completely replace complex biopsies to predict who will survive immunotherapy?
It is a very distinct possibility. The liquid biopsy of the future might just be checking if your artillery is in place.
That is something to think about. If you were looking at clinical trial data in the next few years, don't just look for T-cell counts anymore. Keep an eye out for B-cell panels and IgG1 titers. The T-cell has had its decade in the sun. The B-cell is finally stepping into the spotlight.
It certainly is.
Stay curious and keep diving deep.
Goodbye.